So I want to thank Karen and Eric for inviting me to talk to you today. I am talking on a topic that may sound like Greek to some of you. My topic is HLA Class II antigen presentation, insights into the pathway in health and disease. So what I'm going to do is I'm going to first talk about antigen presentation, the nuts and bolts of antigen presentation, and then I'm going to talk a little bit about why that might be relevant to those of you who are trying to think about new and better ways to treat glioblastoma. And then I'm going to talk at the end about my own work, which is actually on autoimmunity, and who knows what kind of connections that might eventually make for somebody. So antigen presentation is the term that's applied to the way that T cells can recognize protein antigen. And unlike B cells, which can recognize a globular protein using surface antibody, T cells require that protein antigen get processed inside an antigen-presenting cell, and that antigen-presenting cells include dendritic cells, macrophages, B cells. So the protein needs to get processed into peptides, and those peptides need to get presented on the surface of the antigen-presenting cell, bound to an HLA molecule. So HLA is the human leukocyte antigen, which is the human form of the major histocompatibility complex. And that complex is then recognized by a T cell receptor, which has dual specificity for a particular peptide and a particular HLA molecule. So this paradigm of how T cells recognize antigen, peptide, and MHC applies both to cytotoxic CD8 cells and to helper CD4 cells. But there's a fundamental difference. The cytotoxic T cells recognize peptide when it's bound to an MHC class I molecule or an HLA class I molecule. And the CD4 cells recognize antigen bound to an HLA class II molecule. And in both cases, the co-receptor molecule that defines, for example, the CD8 cells, the CD8 protein itself actually binds to the MHC class I molecule. And here the CD4 protein, which is expressed on the T cell, actually binds to the class II molecule. So we've been particularly interested in antigen presentation by class II molecules, which I've just told you are presenting molecules for CD4 T cells. And you'll see in the course of the talk why I've been interested in this particular branch of antigen presentation. But one of the reasons is because CD4 T cells really are the orchestrators of the adaptive immune response. And that's illustrated first here. So up here is this CD4 T cell interacting with an antigen-presenting cell that's expressing class II and peptide. And that CD4 cell can then become any one of a number of effector-type T cells. These are all CD4 T cells still. They have a lot of different numbers. They are polarized by the cytokine milieu in which this interaction with an antigen-presenting cell takes place. And that leads these different cells to have different effector mediators and different effector functions, everything from suppression of inflammatory responses to driving inflammation, a couple of them, and even killing infected cells. We think of CD8 cells as the killers, but there is a branch of the CD4 lineage that also is capable of killing a target cell. So in addition to all those different CD4 functions that come out of the antigen or that follow the antigen presentation event, activated CD4 T cells provide help to other parts of the immune system. So they provide help to B cells. So here's a CD4 cell, and right in the middle there is this little interaction that I focus on of MHC class II and the T cell receptor. And there are other surface interactions, but the interaction between those two cells leads to the generation of a series of cytokines which drive B cell development, B cell expansion, I should say, and affinity maturation of the immunoglobulin. And these CD4 cells over here are, again, interacting with an antigen-presenting cell. Here's this specific interaction between class 2 and the T cell receptor, and this activated CD4 cell is providing help to CD8 cells. And that is occurring both through surface interaction between surface molecules and also the generation of, in this case, IL-2, a critical cytokine for the proliferation of CD8 cells and for their activation as effector cells. So it's for those reasons that I think about CD4 cells as the orchestrators of the immune response, and that's what's led, among other things, to my interest in how they get activated by MHC class 2 antigen presentation. So why would that be of interest to all of you? And I suspect I'm actually showing a slide that really taught me something, but is all old news to all of you, and it just has to do with the fact that there is so much energy and effort now being placed into harnessing the immune system, and in particular T cells, to treat cancer, including glioblastoma. And there are these various modes of doing that. The cellular mode, which includes CARs, as Dr. Brem mentioned earlier, as well as adoptive T cell transfer. That means you've first taken the T cell, you've gotten it activated by a particular antigen, and in glioblastoma, because there's evidence that the tissue includes or expresses CMV antigens, even though the brain tissue around it does not, you can actually, or there are people trying to use anti-CMV T cells in the treatment of glioblastoma. There's also immunomodulatory therapy, and I'm going to just talk about that a little bit more because antigen presentation is really the key partner to these various immunomodulatory therapies. And that's illustrated on this next slide. So here is, again, this interaction that I hope is now familiar to you between an antigen-presenting cell and a T cell and the presentation of peptide. But what you can see is that in addition to what we call signal 1, the antigen presentation interaction that I've just described, there's a need for signal 2, a second signal, to actually get this cell activated. And that second signal occurs through what we call co-stimulatory molecules. In the course of a T cell activation program, there's some negative feedback that's programmed into the process so that the T cell activation is not unlimited. And that leads to the expression of an inhibitory co-stimulatory molecule like CTLA4 or like PD-1. And so the immunomodulatory therapies interrupt that inhibitory co-stimulation. I won't say we really understand how they work. I just heard Jim Allison talking yesterday, and I would say we still don't really understand how these immunomodulatory therapies work, but they're thought to be working, at least in part, by taking the brakes off T cells, that is allowing this, instead of having inhibition, allowing T cell activation to occur. But I wanted to just also point out that that's all in the context of this key partner interaction, which is antigen presentation to the T cell receptor. Of course, vaccination is another approach that also relies on antigen presentation. So here are some of the antigens that are being used in the treatment of glioblastoma, tumor-associated antigens like the EGF receptor variant 3, as well as that's the tumor-specific antigen. Tumor-associated antigens, which include a whole range of tumor-associated molecules, they are not so specific for just glioblastoma, and sometimes there's a problem with the fact that normal cells express these antigens as well. And then there are people using the whole tumor cell lysate. In the vaccination approach, you can either take an antigen-presenting cell and in vitro feed it with antigen, and then give the patient a dendritic cell that's been fed antigen. So this process of getting to the HLA molecule with a peptide goes on in vitro, and that's what you give the patient. You can also give stimulate the dendritic cells in vitro with a peptide in particular. You sort of bypass the antigen processing part. So those are different ways that you can create a tumor vaccine. But there are tumor vaccines being tried in glioblastoma where the patient is actually getting the antigen the same way you would for a vaccination for the flu. And the whole antigen presentation process is going on naturally using the immune system's natural ability to present antigen. Okay, so now I'm going to switch a bit, and I'm going to first go into some detail, hoping that I've convinced you that antigen presentation and a discussion of antigen presentation belongs in this bigger discussion of glioblastoma. I'm going to switch and go into some detail about exactly how HLA class II molecules get loaded with peptide. That's the work that my particular lab has been doing over the last numbers of years. And then I will talk about class II molecules as risk factors for autoimmune disease, which is why I started to get interested in antigen presentation by these molecules. So that's probably the farthest away from glioblastoma, but as I said, you never know where a connection will be made. Okay, so this is the human major histocompatibility complex, the genetic region. And what you can see is that there are antigen-presenting molecules, class II antigen-presenting molecules that come in three flavors, DR, DQ, and DP. They're all encoded by tandem genes for an alpha and a beta chain, because each class II molecule is a two-chained molecule. And then also within this region, and therefore sharing some nomenclature, and also sharing nomenclature because there's some homology genetically and structurally, between these cofactors. There are these two cofactors, HLA-DM and HLA-DO. And DO is the DM inhibitor, and although we've done some work on that very recently, I'm not going to talk about that today. We're going to focus on DM and its function in this loading of peptide. So here's a schematic of how HLA class II molecules get loaded with peptide. So these molecules are synthesized in the ER. They are transmembrane molecules. And very rapidly after synthesis, they interact with a dedicated chaperone that Karen knows a lot about that's called the invariant chain. And it exists as a trimer in the ER. And that trimer acts as a scaffold upon which class II molecules get assembled because they're two chains and they need to be a dimer. So this chaperone molecule helps them assemble and directs their folding. There's also a piece of this invariant chain that sits in the peptide binding groove of class II, so it blocks these molecules from binding any ligands while they're in the ER. And then the invariant chain also has sequences in the cytoplasmic tail that direct this oligomeric complex ultimately to endocytic compartments. And the endocytic compartments contain proteases, and they are have acidic pH, and the proteases are more active at acidic pH, and the invariant chain gets degraded under the action of those acid-active proteases in this very stepwise progression until what's left is just a piece of the invariant chain sitting in the peptide binding groove. It's actually a set of pieces that are called a nested set because they all have the same middle sequence and they differ on either on the N-terminal and C-terminal ends. They're a bit ragged, and that probably has to do with how the proteases nibble them down. So it's actually, we refer to it as a peptide, but really it's in the natural setting, it's a set of peptides that are all very similar to each other. So we refer to these as CLIP for class II associated invariant chain peptide. And CLIP sits in the peptide binding groove of class II. So if this class II molecule is going to be able to present antigen, self or foreign antigen, the CLIP peptide has to be removed and exchanged for antigenic peptides, which are generally also being generated in the endocytic compartment by those same proteases that were processing the invariant chain. So in order for CLIP to be removed and antigenic peptide to bind, for most class II alleles, that requires an interaction with this cofactor HLA-DM. And what DM does is that it binds to an exchange-prone conformation of class 2. It's sort of more open, which allows CLIP to leave. That stabilizes, DM also stabilizes this open conformation. And then, as I'll show you in the next slide, DM has a lot of influence on which of these available peptides actually is the one that gets presented at the cell surface. That's up there. So that last function, which is really very critical, last function of DM is called, we would call it peptide editing. And what that means is that DM interacts with class 2 CLIP complexes, and peptides that don't bind tightly are released until a tight binding peptide binds to the class 2 molecule, and that we believe nucleates a conformational change, a kind of compacting of the class 2 molecule around that high affinity or tightly bound peptide, and then that drives the dissociation of this class 2 molecule from the DM cofactor, and that allows this molecule to go to the cell surface. So these DM-resistant peptides, as I just said, are high-affinity peptides. That means that the cell, the antigen-presenting cell, has this selective system so that the complexes that go to the cell surface are tight complexes that presumably will stay in place for a period of time. And evolutionarily, or teleologically, that makes some sense because your dendritic cells contact antigen at a place and time that is removed from where they have an opportunity to interact with T cells. You may bind antigen out in the periphery, then that dendritic cell has to move to your nearest lymph node, and then it sits there while T cells are circulating through the body. So this selection for tight binding complexes presumably has facilitated better host defense against pathogens. Okay, so that's the diagram of how class 2 molecules, or what we currently think of as how class 2 molecules get loaded with peptide. One of the things that my lab does is work on putting more molecular detail into that picture, and there is more molecular detail, but I'll spare you. But what I will now talk about for the last few minutes is how I got interested in these molecules in the first place. And that is the fact that among the autoimmune diseases that particular HLA alleles, HLA, as I'm sure you all remember from medical school or from somewhere, HLA is the most polymorphic locus in the human genome, so many, many different alleles, but particular alleles, inheriting particular alleles, predisposes to particular autoimmune diseases. So if you inherit HLA-DQ2, you have a 250 times greater risk of getting celiac disease than compared to somebody who doesn't have DQ2. Okay, so we were very interested, or I became interested really during my pediatric rheumatology fellowship many, many, many years ago. Got interested in why it might be that particular alleles would predispose to particular diseases. And one of the reasons that we studied just how HLA molecules get loaded with peptide was we figured in order to understand something as apparently complex as that, we were going to need to understand in molecular detail how class 2 molecules get loaded with peptide. And in fact, it does look like we need to understand that level of molecular detail. Our hypothesis was that once we had worked a lot on the pathway, was that the disease-associated alleles might navigate that pathway differently than a non-disease-associated allele. And let me check the time. Where am I? Okay, that should be just about right. Okay, great. Okay. Thank you. Okay, so what we decided to do was compare closely related alleles that differ in terms of their association with rheumatoid arthritis. And when I say differ, I'm really only talking about 3 or 4 amino acids, although probably placed at critical positions to influence the overall function of that molecule. But the difference between these disease-associated alleles and this non-associated allele is 4 amino acids. So why was that causing a difference in disease association, or why does that? We still don't really know the answer. But the Namrata Patil, who worked on this as a postdoc, compared a lot of things that you might think of comparing if you thought about that whole diagram of how things happen. And I'll just go to the punchline for what she found was different. what she found was that the disease-associated alleles lose that CLIP peptide spontaneously. They don't need the help of that cofactor HLA-DM. And that's shown here by, this is a pulse chase experiment where you metabolically label cells and then you immunoprecipitate the class 2 molecule. And interestingly, the CLIP peptide, because it's very methionine rich, if you've done a S35 methionine label, you can actually see that peptide on this 1D gel. In any case, what you can see is that the disease-resistant allele over, these are days here, is keeping hold of that CLIP peptide, and that's in a cell that has no DM to kick it out. Whereas in the same cell that has no DM to kick out the CLIP peptide, these two disease-associated alleles are losing that peptide anyway, and that's like the loss of that band. So that was the key biochemical feature that she found in these disease-associated, in this set of disease-associated alleles, which is the rheumatoid arthritis association. This is I'm going to skip this one in the interest of time. It's just recombinant molecules showing the same thing that we saw in a cell. Okay, so that would be sort of just a biochemical curiosity, but it turns out that a number of alleles that are disease-associated for different diseases have this same phenotype of having low affinity for the CLIP peptide so that they lose that peptide spontaneously and they don't go normally through this whole editing process that I told you about. And that's true for the disease-associated allele for IDDM, for type 1 diabetes, both in the human and in the mouse model, as well as some other even rarer diseases. So we wanted to understand, well, what is CLIP affinity? What is that doing for antigen presentation in the cell? How would that have some kind of, or what kinds of effects does that have? And in order to try to answer that question, Cornelia Rinderknecht, who was at that point a graduate student in the lab, said, okay, well, I'm going to take this area of the invariant chain where the CLIP peptide comes from, and I'm going to make some mutations there that make that affinity tight again so that these alleles now are holding on to CLIP because the CLIP itself has been mutated, and say, what does that do in the cell? How does that change the function of that cell? And I'll go through this somewhat quickly instead of really going through all the nitty-gritty of the data, and just tell you that if you have normal affinity, really, for CLIP, you have more cell surface class 2 than if you have low affinity. So the disease-associated alleles have low affinity for CLIP, and one consequence of that is they just have less class 2 at the cell surface. That's one consequence. Another consequence is that the half-life of class 2 molecules is longer if you have better CLIP affinity. And remember, the disease-associated alleles have poor CLIP affinity. So I'll just show you her experiment. This is with an allele that has low affinity, and this is a Western blot where we've blocked new protein synthesis, and we're looking at the class 2 molecule over time after we block new synthesis, so we really are looking at turnover in a simple assay with a Western blot. Anyway, you can see that over time that molecule is going away, whereas if we express this molecule in a cell where we've made the CLIP peptide have high affinity for this class 2 molecule, then the class 2 molecule is much more stable. It's still present and strong in high abundance at 22 hours. And then the next two lanes are controls. Okay, so now I've shown you that having good CLIP affinity does two things. It leads to a longer half-life of this class 2 molecule and more on the cell surface. And the other thing that having higher CLIP affinity does is that it reduces the ability of the class 2 molecule to undergo peptide exchange at the cell surface. And again, I think in the interest of time, I won't really go through this except to say that this is a T cell assay, and we're looking at how the T cell is getting stimulated by a class 2 molecule. And if we have increased the affinity for CLIP, the affinity of CLIP and the class 2 molecule, then we don't get good peptide exchange on the cell surface, and so we don't get good T cell activation. Whereas in the disease-associated alleles, there is better peptide exchange at the cell surface because the clip molecule falls off. The molecule does not go through that editing, and therefore it's easier to exchange what's at the cell surface with another peptide. Okay, so what's the link to autoimmunity? I've told you that a number of class 2 alleles have this low affinity for this clip peptide, and we don't know yet exactly what the link to autoimmunity is. These are our hypotheses, and we're testing them in animal models where the animal has a class 2 molecule with low affinity for clip. But I've told you that this low affinity for clip leads to low class 2 levels. We think that may reduce the efficiency of central tolerance for the immunologists in the crowd. And then the thing that I think maybe is more obvious even for a non-immunologist is that if you can exchange peptides easily at the cell surface, that will allow that class 2 molecule to present new epitopes that are being generated at sites of inflammation. And those new epitopes, even from self-peptides, may be epitopes that you have not in any way tolerized against. So that could be a way that you would get to autoimmunity, that these new epitopes that are generated in the context of inflammation by proteases in the inflammatory site can much more easily exchange on the cell surface and be presented to T cells that can recognize them. Okay. Now there was one allele in the group that are associated with common diseases that didn't fit our low affinity for clip model. And that's DQ2, which actually turns out to have a low affinity directly for DM, the cofactor. And that's an allele that's associated with celiac disease, also with type 1 diabetes, with juvenile dermatomyositis, with systemic sclerosis. Here's the key piece of data that shows that this DQ molecule has low affinity, or has, this is actually the first of two pieces of evidence that you have to put together to see that this molecule has low affinity for DM. So what these are traces from a mass spec of peptides that you can elute from a class 2 molecule. So class 2 molecules bind peptides, you can purify the molecules, elute the peptides, and run them on a mass spec. And if you take a molecule that has normal interaction with DM and normal interaction with CLIP, if you express this molecule in a cell that has no DM, and you purify the molecule and elute the peptides, the only peptides that will be there are CLIP peptides because there's been no DM to remove them. Whereas if you express that same molecule in a DM expressing cell, then you have a whole forest of different peptides because DM has removed the CLIP peptide and a lot of other peptides have been put there in the groove in its place. The DQ2 molecule, surprisingly, whether it's expressed in a cell with DM or without DM, has a very similar pattern of the peptides that you can elute. And the primary peptides are CLIP peptides, whether DM is there or not. So that could either tell you that CLIP peptides have very high affinity for DQ2 and DM can't even remove them, or that DM has low affinity for DQ2 and it doesn't remove the CLIP peptides. And the second turns out to be the case, and I will not go through the data except to tell you that it's the second, because I think I'm too close to the end of time. Yeah, so it is the second that the DQ molecule has low affinity for DM, so the CLIP peptides do not get removed. So what we're able to show with this, using this molecule in an antigen presentation assay, is that if we took T-cell clones from the guts of individuals, from the small intestines of individuals with celiac disease, they, as you may know, those T-cell clones have a reactivity to gliadin, and that's what sets celiac disease off. And if we did an antigen presentation assay where we overexpressed DM in order to overcome the affinity barrier between DM and DQ2, what we found in the white bars here is that the presentation of gliadin was suppressed, sometimes down to nothing, when DM was overexpressed so that you could actually make DM interact with DQ2. And what that means is that, I'm sorry, let me just go back here for a second. What that means is that if DM and DQ interacted normally in the intestine of an individual with with propensity or possibility of celiac disease, this interaction would remove the presentation of the gliadin peptides, which is what drives that disease. So the link to autoimmunity here, we think, is that low DM affinity of DQ2 allows these gliadin peptides to be presented, and they would otherwise be edited out or prevented from binding. So that brings me back to this is my last slide, and that just brings me, except for my acknowledgments, and that just brings me back to this issue of what is different about disease-associated class 2 molecules compared to disease non-associated class 2 molecules. And I guess I've sort of rushed through, but the bottom line here is that the difference is all, I wish I could get this to really stay lit. And okay. Try this. Okay. Is that the difference is all right here. Either the disease-associated allele has low affinity for CLIP and just spontaneously loses it, or the disease-associated allele has a problem interacting with DM. And in either case, the disease-associated alleles are not going through this quality control process of peptide editing that leads to these tight binding complexes on the cell surface. So we think that's a critical aspect of why particular alleles predispose to autoimmune disease, because they really are escaping what is this critical process. Now, one thing you would ask is, well, why would alleles that are escaping some critical process still be in the population? Why would evolution not have just gotten rid of those alleles over time? And that brings me to, or that leads me to remind you that the thing that drives evolution, or that we believe has driven evolution of the immune system, is dealing with pathogens, not dealing with cancer and not dealing with autoimmunity, which is probably one of the reasons why it's as hard as it seems to be to harness the immune system to deal with cancer, because that's really not what it evolved to do. It evolved to deal with pathogens. And so we believe that these alleles that predispose to autoimmunity are still in the population because they provide some advantage for some pathogen that the human species either still has to deal with or had to deal with during some time in our history. And having alleles that get to the cell surface in a way that can be easily exchanged for other peptides, et cetera, presumably provided a survival advantage at some important time in evolution, or maybe even now with pathogens where that's a critical part of the defense. The last comment I'll make is that you might say, well, if all the alleles have these same problems, why do they lead to different diseases? And there are probably two answers to that. One is that although these factors are the strongest genetic factors for autoimmune disease, in any autoimmune disease, they're not the only genetic factor. So the particular disease may be influenced by what the other inherited risk factors are. And the other thing is it does look like particular peptides are relevant here. And so these different alleles also present different peptides, and that may be part of why you get celiac disease with one allele, type 1 diabetes with a different allele, rheumatoid arthritis with a different allele. Okay, so that is the last comment. And I just want to acknowledge people who have done this work, and I've mentioned some of them along the way, but these are all really talented PhD and MD PhD students in my lab who have helped us figure out at least as much as we figured out so far about disease and alleles and autoimmunity, and these are some of our collaborators, and I thank you all for your attention.